But before that, we have a master's student at the University of Northern Colorado going to present to you on a member of the Western Rattlesnake Complex, and it's going to be Mr. Calvin Vick, and we're really excited to have him here today talking about some of his venom research. So give it up for Calvin.
All right. So for my talk, we're going to do one of my favorite things, which is leave Colorado. No, just kidding. But I mean, for herb stuff, Arizona's pretty fun. So yeah, I looked at venom variation, and I'm currently looking at venom variation in the Arizona black rattlesnake, Crotalus cerberus. And I want to start by breaking down the title a little bit, and starting with venom. You all know venom, the yellow stuff that comes out of rattlesnakes. But just kind of like a generic definition of it, it's an evolved trait deployed to interfere with homeostasis of a receiving organism, and the organism that's giving it, like a rattlesnake, or say a platypus, which maybe didn't know is venomous, might do it for digestive purposes, defensive purposes, or purposes of prey acquisition. But for this talk, I think you should really hone in on the digestive purposes, because that's something that might be important later. Venom is something that's evolved over 100 different times in the animal kingdom, so it's pretty ubiquitous. And then just kind of generically what it is, a combination of peptides and proteins. And then what it's studied in, many different animals, but there's a big emphasis on snakes, I think, because snakes are super cool, but there's something we definitely should not forget, and that's around the world snakes are killing over 100,000 people. In the US, that number's 5 or less, but it's very serious elsewhere. So the second part of my talk, venom variation, and what exactly that is. So obviously different species have different venoms. The cobra bite's going to be different than a rattlesnake bite. But then there's also this aspect of intraspecific venom variation, and that's just where you're getting venom changes within a single species. So I've got a figure up here from a paper that came out last year by UNC's Dr. Kara Smith, and she looked at the prairie rattlesnake and the way its venom changes across its range. The species goes from Mexico all the way to Canada, and of course they're eating different things all the way across their range. And so she found somewhere around the I-70 corridor there's a major split in the venom phenotype, with ones further south being more hemotoxic, and then ones further north being more neurotoxic. And so a couple of reasons that we think we see this interspecific venom variation is ontogeny. So as an organism, as a rattlesnake kind of grows up, it's changing what it's eating. So it's really common to see a rattlesnake when it's younger eating things like lizards, frogs, and then as it grows, switch to a more mammalian-based prey, just larger, more calories. And so those prey systems obviously have different physiologies and things like that, so this venom kind of changes with that. And then the second role, maybe you've seen some of the work that came out of UNC prior, looking at resistance. So a lot of prey items will become resistant to whatever the local venomous snake is. So in the case of the prairie rattlesnake, the prairie rattlesnakes you find in this area are going to be are going to have a tougher time killing their prey than, say, a snake from elsewhere, and that's just because that prey is becoming adapted to it. So just kind of briefly mentioning too, there's also a role of venom variation in snake bite, and just that it adds kind of some complexities to it and the manufacturing of it, and can change the efficacy of snakebite treatment. So the last part, the Arizona black rattlesnake, I'm sure many of you are pretty familiar with it. Obviously it's not found in Colorado, but it is endemic to the United States. It's found in just Arizona and New Mexico, and fairly specifically along the Mogollon Rim, which is a geologic feature that kind of splits Arizona in half. It's part of the Grand Canyon. It's the reason you see those really tall and cold mountains, and the reason Arizonans are able to ski in the winter. So it's pretty interesting and extremely varied. And then obviously that's what the Arizona black rattlesnake looks like. But they're really variable. They're a large-bodied rattlesnake, and they are medically significant. So, kind of the reasons we were thinking this species might experience quite a bit of venom variation. One being they're montane, so for the most part they're mountainous, but if you look at the mountains specifically in which they're found, there's a huge elevation gap. I mean, I think the record is somewhere around 9,500 feet. And not going down to sea level, but they're not too far outside of Phoenix, and if you've been in that area, you know it looks really different. The rattlesnakes really far west, and the cerberus and the walleyes, really dry, arid, compared to some of these high elevation forests they're kind of characteristically thought of being found in. They're also a member of the Western rattlesnake complex, which is a pretty complex just in being that there's not really good species boundaries, and so you get a lot of potential for influence in just being hybridization within different members of them, and you see that a little bit here in Colorado. If you've ever been down to the Four Corners region, you see the rattlesnakes down there. They're super weird, and then as you go north, you start to get this weird concolor influence before they turn into what would be fully concolor. So another reason is there is some genetic variation within the species. So a paper in 2016 published by Douglas et al., they looked at population genetics, and while they lacked some of the far western populations like the cerberus and the walleyes, they kind of hit most of the central range areas, and they found probably about 5 clades, judging what they broke it up into, so at least there's evidence of isolation, which can then lead to some sort of runaway selection, or at least just they're not mixing their venoms as much. And they also found that these clades kind of followed rivers, some of the major rivers within Arizona. So yeah, just another reason that this species might have some sort of variation going on. And then briefly talking about the previous work, so my advisor, the esteemed Dr. Mackesy, published a paper in 2010 looking at the entire clade, CVOS, sometimes referred to as, or I guess in this case just the Organus members, and he noticed a trend which he labeled the type 1 venom versus the type 2. So the type 1 snakes are what we call tenderizers. These are the snakes that are really hemorrhagic. They're going to break down a ton of tissues. The hemotoxic snakes, and they're loaded with all these different enzymes that are breaking down proteins. And then the type 2 snakes are your really toxic snakes. So species like concolor, they just have really good killing power. They're really efficient, and you definitely see it in how they're taking down prey. And if you look at this figure, sorry you guys over there, but if you look at this figure, the CO on the bottom represents SVMP levels, which is snake venom metalloprotease, an enzyme really important in breaking down proteins, a strong indicator of type 1 snakes. And so the concolor you can see has almost no activity levels, where CE is cerberus, and it's got the highest activity levels of any of the species. However, this paper used 1 snake per species, which then limits the sample size and locality. But at least from previous research, it's looking like these snakes are really hemorrhagic and clear type 1 snakes. And so my project is then telling the whole tale. Notice the spelling for cerberus. So to contextualize it a little bit, a little morbid, yes. So let's see. You guys left. There's a picture of a person who was bitten by a snake that we would call type 1, and so you can just see there's extensive tissue damage there. And then on the right, there's a child experiencing something called ptosis, where he's losing muscle function. He's unable to keep his eyes open. And this is from a South American rattlesnake, Crotalus, which is a type 2 rattlesnake, and very scarily so. So how do we do this? So my favorite part is the catching of the snakes. I'm a big field herper, and so I've been able to spend a ton of time over the last summer going out and catching cerberus in different places, but sometimes we can't do that, and so we'll contact other people who might keep the species, and as long as they have locality data, we've included them. Snakes are then extracted. Sometimes we do this in the field, sometimes we do it in the lab. I've got a picture of Dr. Mackesy extracting a Zobcon rattlesnake in the lab. It's very nice. And then I've got another picture of 3 cerberus knocked out in a Motel 6 bathtub. So there's some levels to it, and I don't have pictures because I was alone, but I've also done a fair share of on the side of the highway extractions, and that's, yeah, pretty interesting. So, but how we do it is we anesthetize the snake with something called isoflurane, and this just minimizes the chance of risk for the person extracting the snake, and then the snake hurting itself, because they tend to flail a bit when you pin them down. So when you do that, they sleep nicely. So how are we determining these activity levels? We're doing assays for 6 enzymes that are extremely conserved within rattlesnakes. And I've got them labeled here, and I'll go in a little bit into more detail about them, but just briefly what they are. And then so kind of the procedure, the assays all have levels of variation in conducting them, but the formula is kind of the same. So you mix your sample of venom, which is at a set concentration, in with a buffer, which maintains a pH, usually around 8, and that's because Dr. Mackesy has found previously that venom activity changes with pH, and 8 seems to be about the optimal activity for it. You then mix it in with a substrate, which has a binding site that mimics where that enzyme would bind on, say, your blood or something like that. You allow it to incubate. The sample is then binding to that substrate or cleaving something there, which causes like a sort of color change, cleaving a chromatophore, and then using a spectrophotometer, you can read the changes in absorbance from those being released, and then using some equations, you can figure out specific activity and get a rough idea on how much is there, or at least how active it is, which then is a proxy for how much is there. So far what have I found? Well, at least in terms of sampling, I've been able to get 35 snakes, although we've got some more incoming, and I've got a bad figure from Google Maps, kind of where I've been able to sample, and then looking at those clades found, you can see that I've covered all the clades, although I just don't have sample sizes for each of them that I'd like to have, but some of those that might not be rectified in some of them just because I'm trying to graduate. But you can see for the most part though, we've pretty much covered them across the board. There's some holes, but what have we found then looking at the venom? So kind of as expected, SVMP activity remains fairly high across the board. The one that's labeled 1 gray, those are snakes from the far eastern population, so some low levels there, although these were some smaller snakes, but across the board, pretty much we are seeing high levels of SVMP activity. Then PLA2 activity, this is an enzyme that's just generically cytotoxic, so it's just causing cell destruction. Fairly low levels, something seen in a lot of rattlesnakes, but there is some level of variation across populations. Oh, and the colors, I tried to code them with the Douglas et al. Clades, but then the black on the far side is just the total average. So then 2 serine proteases, we've got thrombin-like, and so this is going to cause or prevent your blood from clotting, and so fairly low activity, kind of something I was at least anecdotally told we've seen before, but then really interestingly, populations from the cerbats or wallopis, sometimes referred to as wallopiensis, they show really high levels, which is really interesting, and then that green population, which is just kind of central Mogian rim, they're showing really low levels of thrombin-like, and then the other serine protease, callicrin-like, you can see this population then takes off in it, while this one drops to close to the average, and so it almost looks like there is some sort of trade-off there. Oh, and then callicrin-like is involved in vasodilation, so just dropping blood pressure. Then we've got The last two of the 6 enzymes, phosphodiesterase, so this one is just involved in destroying secondary messengers. Not super important and not really well understood, but just pointing out that there is some level of variation there. And then LAOs are involved in cell death as well. And so, again, not the most important, but we're seeing some levels of variation in these populations from far eastern Arizona and New Mexico are really interesting in that they have pretty much zero activity in it. And a fun fact about this one is that it's the enzyme that causes the yellow coloration in venom. And so snakes from Arizona pretty much have clear or white venom. So what can I say about the snakes that I've been able to sample so far? High metalloprotease activity, kind of what we expected to see. They seem to be clear type 1 snakes. Some of the other enzymes have revealed there's not the neurotoxins you see in type 2 snakes. We've got some weird levels of variation in some of the other enzymes. Snake venom metalloproteases are high in the west and in some of the central populations. Low LAO activity in the east. It's also revealed some areas we need to sample more. And then briefly, my study doesn't really answer the evolutionary ecology question of why type 1, but I still like to think about it. And so kind of what we're thinking is that this venom is really useful in digestion. For the most part, these are high elevation snakes. They're black bodied, which tends to correlate with their living in colder temperatures. It helps them heat up faster. It helps them digest the dark coloration, and so maybe this is just another step in aiding in digestion in cold places, because if you've ever been to like where their peak is, it looks like you're in Estes Park or something. It's full of aspen trees and pine, and it's cold. It's 40 degrees at night in the summer. It's cool. But yeah, and then with that, I'd like to thank everyone in my lab, Ryan, of course my PI, Dr. Mackesy, all the people that have given me funding, and I think I'm pretty much out of time and overtime, but I'll still take questions maybe.
One question if anyone's got one for Calvin.
Nice presentation. I was interested in the fact that you included captive snakes, because you brought up earlier in your presentation that genetic effects might influence venom characteristics.
Yeah.
So wouldn't the captive snakes, depending on how long they've been in captivity, not have those effects? You're just feeding them like pinkies and mice, so they don't see that variation in diet.
For the mobile, so it's something that actually like our lab's going to go into this week, but it's not super well understood, like the effects of captivity on snake venom, and so right now we're just going to have to assume it doesn't, but it's in some of the papers that have been published on it, like, are back and forth, and some show maybe a few snakes are affected by it and some aren't at all. So that's kind of what we've got to play with right now.
If you got more questions for Calvin, we're going to a break in just a second here. But Calvin, what's the story with that photo?
It's Ryan on the couch. We're just hanging out.
All right, just wasn't sure. All right, well, thank you, Calvin. We appreciate your time.